Today we have a presentation by Mei Iwamoto. Iwamoto is a biological research technician with the Hawaiʻi Cooperative Studies Unit. Her presentation is titled Stairway to Extinctions, Documenting the Use of Stream Habitats by Mosquitoes in Hawaiian Forests. Her research is conducted through a permit with the HETF, and please note that permits are required when entering the HETF for research purposes. Mei, you may start whenever you're ready.
Okay, awesome. Let's get started. Thank you to Tristan for that great introduction, and also to the Hawaiʻi Experimental Tropical Forest for organizing this webinar. I'm really excited to be presenting here today. My name is Mei Iwamoto. I'm a technician at the Pacific Islands Ecosystems Research Center, and I just want to take a moment to shout out all of my fellow co-authors on this project. Who work on this project with me, and many of whom offered really great feedback for this presentation. So I really appreciate that. And so today I will be presenting this talk titled Stairway to Extinctions, Documenting the Use of Stream Habitats by Mosquitoes in Hawaiian Forests. And while our title might seem a little bit of alarmist, it does underline a really possible uphill invasion route for mosquitoes into high elevation forest bird habitats. That are still relatively disease-free. And that's just going to be the topic of today's talk. First, I kind of wanted to go over a brief outline of what this talk is going to be about. I'm going to begin by giving some brief background information. And for folks who may not be as familiar, I'm going to be starting off by talking about Hawaii and our native forest birds, as well as their recent population declines. And then I'll be moving on to briefly introducing avian malaria and mosquitoes, as well as malaria transmission and how climate is beginning to affect that. Next, I will segue into some of our research questions for this project, as well as the data collection and field work that we've been doing to answer those questions. So that includes things like larval and mosquito monitoring, as well as counting habitat abundance in our Hawaiian streams. And our fieldwork has been taking place at Hakalau Forest National Wildlife Refuge, as well as the Laupāhoehoe section of the Hilo Forest Reserve. And finally, I'll be presenting on some of our preliminary findings that we have so far, as well as some of our takeaways from those findings. So first off, I'm sure most, if not all of us, are very familiar with the Hawaiian Islands, but if not, the Hawaiian Islands is the most isolated archipelago. We have these relatively large and ecologically diverse islands here. And that, along with its insular nature, makes it a really unique environment, and it's led to a lot of adaptive radiations among our endemic taxa. So some of our radiations, we have some among some of our plant species, as well as other animals. But in the case of our birds, the most famous and amazing example would be with our honeycreepers. So Hawaiian honeycreepers are a monophyletic group of small finches. They're once again a really incredible example of adaptive radiation, where they've radiated into more than 50 recognized species. And radiation in honeycreepers happen first and foremost about food and their bill shape, where we have these shifts in their feeding behavior that lead to selection for changes in bill shape so that they can better take certain kinds of food. And so in this graphic, you'll see all of our birds with that wide variety of bill shapes that they have. And so we will have differences in bill shape here, depending on if they're insectivores or nectivores or seed eaters, for example. So just as an example, we have our EEV right here. I don't know if you can see my laser pointer, but that's our EEV, and they are nectivores. They mostly like to eat nectar. And so they have a bill shape that is intermediate in length and curvature, and it's well adapted to taking nectar from more tubular flowers. Another example, we also have Palilas, who are seed eaters, and they have more of a conical bill shape that makes it really good for them to crack open hard seeds. Finally, we also have some Aequiopolaos, who are actually insectivores. And Aequiopolaos are really interesting birds. They're they've kind of evolved to fulfill a woodpecker niche. And so they use their beak to gouge open wood and look for insects underneath. However, from more than 50 recognized species, we are currently down to only 17 species left. And many of those 17 species of our honeycreepers are endangered or threatened. 4 species that are of immediate concern are the akekee, the akikiki, the akohekohe, and the kiwikiu. And regarding the akekee and the akikiki, their first declines were seen on the island of Hawaiʻi in the early 2000s as species ranges contracted deeper into the Olokai swamp. And today, unfortunately, akikiki numbers have been reduced to a mere handful of birds, and they are functionally extinct in the wild. Similarly, the number of kiwikiu and akohekohe birds on Maui are also in a steady decline. And here are some graphs and figures showing the population sizes and trends for those 4 respective forest bird species. We do have other species on Maui and Kauaʻi who have also suffered significant declines in their population, but these species in particular are of note because of their smaller population sizes, which just further heightens their extinction risk in the near future. And so just something to note with these figures is just their very steep declines in their curves on the graph. And so what exactly is driving all of their extinctions? There are many factors that contribute to their decline, such as habitat loss or habitat degradation, but the general consensus today is avian malaria. Avian malaria is an introduced to mosquito-borne disease. It's caused by a protozoan parasite called Plasmodium relictum, and that reproduces in red blood cells. And so if the parasite load is sufficiently high, the infected bird loses its red blood cells, and those are really important for moving oxygen around the body. So losing those cells can lead to progressive weakness and death in our birds. One thing to note about Plasmodium is that it does have a temperature-dependent development cycle. So malaria is an introduced disease, and many of the Hawaiian honeycreepers are highly susceptible to it, in part just due to the very insular and isolated nature of the Hawaiian Islands that they had evolved in. And the primary vector that spreads avian malaria in Hawaii is the southern house mosquito, Culex quinquefasciatus. It was first observed in Hawaii in 1826, actually on Maui off of a whaling ship called Wellington. And so it is an introduced species, and we actually have no native mosquitoes here in Hawaii. So any mosquito that you come across here in the islands is introduced. So the life cycle of Culex quinquefasciatus, it starts when an adult lays its egg rafts on the surface of standing water. Those eggs then hatch into larvae within a couple of days, depending on temperature, and then they eventually pupate and emerge as adults. And so this larval development cycle for Culex is also temperature dependent, which means that the larvae only continue to develop when temperatures are warm enough. In this case, that critical threshold for temperature is 10 degrees Celsius. So if temperatures fall below that threshold of 10 degrees Celsius, the larval development of those mosquitoes will stop and their survivorship rates drop dramatically. And Culex quinquefasciatus is extremely common throughout the tropics and the subtropics, where the larvae are usually found in stagnant bodies of water associated with human or livestock habitation. In Hawaiian forests, though, suitable larval habitat is limited, but Culex quinquefasciatus larvae will use stagnant pools of water, like in intermittent drying streams, or in hapu'u cavities, or tree fern cavities that have collected rainwater. And so these are some pictures of some real life examples from out in the field. In addition to Culex, we do have another mosquito species that could occur in the similar larval habitats. Here we have the Asian rock pool mosquito, otherwise known as Aedes japonicus. which was detected on the island of Hawaiʻi in 2002. It is now widespread on all the main Hawaiian islands. Unlike Culex, they are better adapted to stream habitats because they have these drought-resistant eggs that they attach to the rock pool substrate. Aedes japonicus, however, unlike Culex, is not a vector of avian malaria. And so most streams on the windward side of the Big Island near Laupāhoehoe have historically been more perennial in nature, which just means that they're typically flowing with water regularly. These streams are also pretty flashy, where they experience these high water events that regularly wash out the stream bed. And so as mosquito larvae are adapted to stagnant water, perennial streams don't really provide good habitat for them. But with intermittently flowing streams that have rockier channels, that could potentially have a number of rock holes that could be suitable habitat. And so just based off of that, there are two components that comprise available larval habitat. The first one being the number of stagnant pools. And the second one is the persistence of those pools. Like, are there periods between those high water flushing out events long enough for the larvae to complete their development? So like I mentioned in some of my earlier slides, both the vector, which is Culex quinquefasciatus, as well as Plasmodium relectum, have developmental cycles that are dependent on temperature, and so they don't develop completely unless temperatures are warm enough. For Plasmodium development, that threshold temperature is 13 degrees Celsius, and for Culex, it's 10 degrees Celsius. And so historically, having these thermal constraints have limited that presence of the vector and the parasite to lower to mid-elevation forests where those temperatures are warmer, which means that disease transmission of avian malaria has also been limited to those areas only. And so typically this meant that the higher elevational areas were disease-free, and the last remaining safe spots for our forest birds who are really susceptible to avian malaria. However, with recent warming trends, malaria transmission may now start occurring at these higher elevations as well. But increasing temperature may only be a part of the problem. Another potential impact that we have with climate change is changing rainfall patterns. When we have less rain and longer periods of drought, our Hawaiian streams that were typically more historically perennial could become more intermittent in flow. And then that could lead to the formation of more larval mosquito habitat that doesn't get flushed out regularly with these high water events. So this leads me to the basis for our main research question, which is how could changes in precipitation and hydrological factors affect the availability of larval mosquito habitat along streams as perennial streams become more intermittent? And then in turn, how might this affect malaria transmission in the high elevation forests of Hamakua? And so with all of that in mind, I wanted to introduce one of the spots where we have some of our field sites at. This is Hakalau Forest National Wildlife Refuge. It is one of those few remaining high elevation refuges for forest birds on the island of Hawaiʻi, and it's located on the high windward slopes of Mauna Kea. It was established in 1985 to preserve some of the best montane forest habitat. And historically, forest bird populations have been pretty relatively stable at Hakalau. Oh, sorry about that. So historically, forest bird populations at Hakalau have remained relatively stable, especially in comparison to their populations on Kauaʻi and Maui, where we are seeing those rapid declines. And so at Hakalau Forest National Wildlife Refuge, we have a total of 4 field sites. 3 of them are along Hakalau Stream. Their elevations are 1750, 1500, and 1250 meters respectively. And we have another field site along Nauhi Stream, which is at 1600 meters. And so since the early 1990s, there have been a number of short-term studies that have looked at the prevalence of malaria in forest birds, as well as the occurrence of Culex quinquefasciatus at Hakalau. In 2012, we found only around 2.5% of amakihi were positive for malaria there. There have been a few detections of individual adult and larval Culex mosquitoes. Those larval mosquitoes were associated with nearby ranching activities, and that habitat has since then been removed. There has been only one detection of larvae in a naturally occurring stream pool. And the adult Culex mosquito detections that I just mentioned may represent occasionally wind-blown mosquitoes up from lower elevations during storms. And so at the moment, there is no evidence of an establishment of a persistent population of Culex at Hakalau, but there is an establishment of the introduced Aedes japonicus with Culex. And the second site that I wanted to introduce, where we also have our field sites at, is Laupāhoehoe Forest Reserve. While there is no evidence of Culex quinquefasciatus at Hakalau, we do have documented establishment of adult and larval mosquitoes at Laupāhoehoe Forest Reserve in 2013. And this forest reserve is adjacent to Hakalau, and it has a very similar elevational gradient there, which makes the site a really good comparison site for our field sites at Hakalau. And so we have 3 study sites along Kauaʻi Stream at Laupāhoehoe, the upper site being at 1250 meters, the middle site is at 1200, and then our lower site is at 1100 meters. And so this leads me to a couple more research questions that we have. First off, how does local precipitation affect stream hydrology and the abundance of available larval mosquito habitat? What is the occurrence of mosquito species in the defined habitat types? What role does water temperature play in the occurrence and persistence of larval mosquitoes? And finally, can streams serve as a pathway of mosquito invasion into Hakalau Forest National Wildlife Refuge? And to answer these questions, we have been collecting climate data like precipitation, ambient temperature, water temperature in the streams, humidity, and stream height at each of our field sites. We have also been doing larval mosquito monitoring and counting the available larval mosquito habitat every month at our sites. So in order to determine the abundance of larval mosquito habitat, we have been regularly sampling the same 200 to 250 meters at every single field site each month. And so that 200 to 250 meter stretch is measured out into 10-meter sections. So for every single 10-meter section, we count the number of standing pools of water there are in the stream bed, and we also categorize them based on size and location in the stream. And here are some of the categories that we use for our rock pools. This is a really good cross-section of what a stream bed might look like. So starting off from the left-hand side of the cross-section, we have our perch pools. These are pools that typically occur high above the typical height of the stream. They're usually fed by surface runoff water. And so because they're located up higher along the stream, they usually don't get washed out as frequently in a high water event compared to the rest of the stream bed. They often have vegetation going along the margins, and they also have a deep substrate of soil and leaf litter. And then moving on, we have our small and medium pools. These are rock pools that are located more so in the stream bedrock channel. Small pools have a diameter of less than 20 centimeters, and medium pools have a diameter of 20 to 100 centimeters. They often have a shallow layer of alluvium as well as some leaf litter. And finally, with our large rock pools, these are pools that have a diameter of over 100 centimeters. They're often at the lowest point of the channel when stream flow has stopped, and they have a deeper layer of alluvium, but they're less likely to have as much leaf litter. And here are some pictures of what those pools kind of look like out in the stream. Here on the left-hand side, we have our small pools that are less than 20 centimeters. The middle picture, it's kind of a good comparison shot of some small pools along with the medium pool at the very bottom. And finally, in the right-hand picture, that is clearly a large pool over there. It's definitely bigger than 100 centimeters. And after we count the number of pools in a single 10-meter section, we then try to dip every pool there is in that section to see if there is a presence or absence of larval mosquitoes. And if we do find any larval mosquitoes, we try to ID them down to species. And this is supposed to be a video of what a dip looks like in one of our pools, and it has some larvae floating in it, but I don't really know how to play it, so I might just skip it for now. This dip, though, did have some Aedes japonicus larvae at different instars, which were the different developmental phases that they have. Oh, yeah, it's playing. You can see them swimming around at the very bottom of the cup right there. Moving on, in addition to counting larval habitat and dipping for mosquitoes, we also have 10 repeat sample pools that are set up at every field site. These are individual pools that are regularly and intensively sampled monthly for mosquito larvae. So for these pools, we do a total of 10 dips, and we count the number of larvae, we ID them to species and to developmental phase per dip. And 5 of these 10 repeat sample pools per site have a hobo tidbit logger in them, and that helps us to record the water temperature of those pools. And in order to collect climate data, we've installed climate stations at every site. So these have data loggers that help to record local precipitation, ambient temperature, humidity data. We also have a stream gauge at every stream site, and that helps us to collect stream height data. Moving on, I'd like to talk about some of the results that we've had so far. This is still an ongoing project and we are still collecting data out there. So these are still preliminary results and we're still in the process of examining the data. First, I wanted to start off with a graph of the rainfall and stream height at our middle elevational site along Kauaʻi stream. So this is the stream that runs through Laupāhoehoe. And so for here, the blue line is for rainfall, and then stream height is in the orange. So right off the bat, you can see that increases in daily rainfall typically correspond to a rapid increase in stream height shortly after. And so when that rain stops, that stream height also drops dramatically. These peaks in stream height, like the ones that I am pointing out, represent high water events that will flush out larval habitat. And this data also shows a little bit of expected seasonality and stream height between the summer months, like here, compared to the winter months. The summer months is categorized by low flow and fewer small high water events, whereas in the winter, we have multiple high water events. Here, we're still looking at the same rainfall and stream height graph from the last slide, but this time it's overlaid by graphs that correspond to pool availability / abundance of the site. So in case these graphs are kind of small for everybody, this graph is corresponding to this part of rainfall and stream height. So going from left to right, this column is for the total number of pools that were counted, the mean number of total pools that were counted per 10-meter section. Then we have our perch pools, our small rock holes, our medium rock holes, and finally our large ones. And similarly, this graph corresponds to this section, and the same for all the other graphs. And so one of the main questions that we wanted to answer is how stream flow affects the abundance and relative permanence of available larval mosquito habitat. So our expectation kind of going into this was that habitat would be the most abundant during the beginning of low flow periods. where flowing water would typically recede to the main channel, and they would leave behind a number of water-filled rock pools. And as those low flow conditions continued, we also expected that the number of available pools would decrease just because small, shallow rock pools might dry up. We also expected the number of perch pools wouldn't really fluctuate as much because their position is essentially pretty high up along the stream banks. However, we saw when we were collecting data that the total number of pools varied surprisingly little throughout the year, and the already uncommon perch pools disappeared by the end of the January spell, which is right here. You can see we don't have any counted here. We can see that they've disappeared by the end of this January dry spell, presumably through evaporation. And it was kind of surprising to see that they didn't really come back even after we had this really high water event and lots of rain in the end of February. And overall, we saw that small rock pools were the most abundant and stable type of pool. And so in the winter/spring, we saw more frequent rainfall that led to increases in stream height and presumably more high water events that could flush out the stream regularly, as well as any larval mosquitoes that might have been in the pools. But in the summer months and into sort of the first couple of winter months, we saw these longer periods of time where there was less rainfall and no significant high water events that could flush out the stream. And these low flow periods could be long enough for Culex quinquefasciatus larvae to develop completely from egg and then to pupa and then emerge as adults, provided the water temperatures were above the critical threshold of 10 degrees Celsius. And so, yeah, we have a span of around 70 days right here, beginning from early May to July. We have this really long span right here in the summer of 125 days. And this is from July to November. And we have this a little bit of a shorter span of 48 days that was right around the very first couple beginning winter months. And here, moving on, next I wanted to take a look at some mean water temperatures that we have from our repeat sample pools. So these are temperatures taken from repeat sample pools at our middle elevational site at Kauaʻi stream, which is the stream that runs through the Laupāhoehoe Forest Reserve, as well as some from our Hakalau stream from the Refuge. And September temperatures are in the blue, while the January temperatures are in the orange. So we saw that the temperatures recorded for September for both sites don't really seem to show a great difference. But you can see that it's really in the January temperatures where there's kind of a difference between our two sites along Kauaʻi and Hakalau stream. And Hakalau stream did have lower mean water temperatures in January compared to Kauaʻi stream. And so, like mentioned in my earlier slides, that critical threshold for Culex larval development is 10 degrees Celsius. And that's noted by the black dotted line along both of these graphs. And so that means that if the water temperature goes below 10 degrees Celsius, the larvae will stop developing and survivorship rates drop dramatically. And so you can see that at our Kauaʻi stream site, in both the September and January months, temperatures were definitely above 10 degrees Celsius. But if you take a look at our middle Hakalau stream site, you can see that in the September month, temperatures were definitely above 10 degrees Celsius. But in January, they were roughly either around 10 degrees or less than that. So I want to move into some observations and takeaways that we have so far from our preliminary results. First, from our mosquito monitoring efforts, Culex quinquefasciatus larvae were found at all elevational sites along Kauaʻi stream, but they have yet to be found along Hakalau stream within the refuge. We did see that Aedes japonicus larvae were more common than Culex larvae in Kauaʻi stream, and they occurred at all elevational sites along Kauaʻi and Hakalau stream. And both Aedes and Culex species may occur in the same habitat, and we have found them both within the same pool of water before. But Culex are typically more common in perch pools and medium/large rock pools, while Aedes are more commonly found in small and medium rock pools. And so we saw that mean water temperatures during the summer at Middle Hakalau and Kauaʻi Stream were approximately around 16 degrees Celsius, which was 6 degrees above that threshold of 10 degrees Celsius. And so at that temperature, it would approximately take around 30 days for the larvae to complete larval development. Winter mean water temperatures at Kauaʻi were significantly cooler at 12.7 degrees, which is nearly 3 degrees above that threshold. So in the winter, the larvae would take roughly around 60 days to complete that development. And if you look back to those long durations of like dry periods of time where we didn't have that much rainfall, we did have that little span of 48 days in the winter between high water events at Kauaʻi Stream, but this still might not have been long enough for a winter brood to complete development there. Next, when we move on to look at the winter mean water temperatures at the Middle Hakalau site, they were at or below 10 degrees Celsius. And this would just not support development of Culex larvae at all. And so this may be one reason why we have yet to see persistent populations of Culex quinquefasciatus getting established at Hakalau. And so just to end my presentation, the survival of Culex quinquefasciatus at higher elevation is driven by the available persistent habitat, as well as the development rate of the immature stages. And that in turn is driven by precipitation and temperature. And so for the moment, our data seems to suggest that the stairway to higher elevation may be still close to Culex. But those general warming and drying trends that we see in the islands may soon change that. It may lead to removing that obstacle for them and allowing increased transmission of malaria at the refuge. Okay. Wanted to do some brief acknowledgements to Pacific Islands Climate Adaptation Science Center and Pacific Islands Ecosystems Research Center, Ecosystems Mission Area for funding this project, and also to our U.S. Fish and Wildlife collaborators at Hakalau Forest National Wildlife Refuge for access and logistical support, and also to Hawaii DOFA and U.S. Forest Service, Hawaiʻi Experimental Tropical Forest for access as well. And also a big mahalo to all of our volunteers so far, Rosa, Maddie, Emma, Alexis, and Lauren, who make this project a success. And yeah, that is it for my presentation. I guess if we have any questions.
Mahalo piha Mei. That was so thorough. I don't see any questions currently in the chat, but if anybody wants to hop in while Mei is still here, by all means, please do. And if not, oh, there we go.
I was just curious, is there a difference in substrate between the two stream systems? Versus Paha?
Oh, that's a good question. Both Hakalau and Kauaʻi Stream have pretty rocky channels. I think if you compare like the different elevational sites along both streams, there are some differences in that, like the upper versus the middle sections of the same stream. It's a little bit different. And I would say we have exactly one field site along Nauhi Stream in the refuge, and the substrate there is not as rocky compared to our two other streams. There's a little bit more of moss and vegetation growing there.
Thank you.
Yeah.
I see James in the chat asked, he may have missed this, but have you been culturing and releasing Wolbachia mosquitoes in some of the mid-elevation sites. I didn't hear you mention that, but
Yeah, to the best of my knowledge, we have not been doing that yet. I'm not sure if there's someone else in the talk who could better answer that question, maybe.
Would you like me to try to answer that?
Yeah, definitely.
Okay. Well, if I don't know how familiar everybody is with the current efforts to introduce incompatible mosquitoes on the island of Maui currently, with the intention of suppressing mosquito populations there. So that's scheduled to happen hopefully in the next month when all permits are in place. Everything else seems to be in order. The Big Island is kind of low on the priority list. I think primarily because we have greater issues with rapid extinction going on in both Maui and Kauai. But certainly part of the hope of this project is that we'll get a better idea at the actual risk of Hakalau being affected in the near future. There's a long history of looking for mosquitoes in Hakalau. We haven't picked up that many. The suggestion is, from what we've seen, is that perhaps they're mosquitoes wind blown up to the refuge. But one of the potential pathways is these streams, and that's really what we're focused on. Hopefully, in a year or two, we'll be able to comment a little bit more on what this means for the future of Hakalau Refuge. And hopefully, by the time mosquitoes make it to the refuge, if they do make it to the refuge, we may have a tool available to deal with that problem. But at the moment, no Wolbachia release is planned for the Big Island.
mahalo, Dennis. Thank you so much. I see Ellison in the chat asked, he may have missed this also, but do both species of mosquitoes carry avian malaria?
Yeah, that's a good question. In Hawaii, the only vector for avian malaria is Culex quinquefasciatus, and Aedes doesn't carry it.
Mahalo, Mei. One more question. Someone asked, can mosquitoes breed in tree ferns?
Yeah, I think I had a picture of some hapu'u cavities in my slides, but I believe we have found some Aedes larvae in hapu'u cavities before. I don't know off the top of my head if I've ever found any Culex larvae in hapu'u cavities, but yeah, they can breed in tree ferns.
And it's more so it needs to have the water, right? That these are not the fern itself, but the cavities that are created. And so that like pig damage leads to mosquito habitat, and it's this cycle.
Yeah, definitely.
I could add that recently Stephanie Miladnik had just completed her master's work. She also worked in Hakalau and Laupāhoehoe, and her focus was looking at hapu'u cavities as larval mosquito habitat. And she was unable to detect any Culex in any of the hapu'u cavities that she came across in Hakalau. But when she moved down into the lower elevations in Laupāhoehoe, it was a frequent observation that both Aedes, japonicus, and Culex quinquefasciatus are in those hapu'u cavities.
Mahalo again, Dennis. Thank you both so much for being able to answer all these questions. And